Applications of microarrays. Gene expression analysis. The predominant application of DNA microarrays has been to measure gene expression levels. In gene expression analysis, RNA is extracted from the cells of interest and either labeled directly, converted to a labeled CDNA, or T7 RNA promoter tailed CDNA, which is further converted to CRNA through the abovine amplification process as shown in figure 3. The methods for labeling of the cDNA or cRNA include incorporation of biotin labeled nucleotide, which is subsequently stained fluorescently labeled streptavidin, incorporation of fluorescently labeled nucleotides during the synthesis, and incorporation of a modified reactive nucleotide to which a fluorescent tag is added later, and a variety of signal amplification methods. The two most frequently used methods are the incorporation of fluorescently labeled nucleotides in the CRNA or cDNA synthesis step, or the incorporation of a biotin labeled nucleotide in the CRNA synthesis step. The labeled CRNA or cDNA are then hybridized to the microarray. The array is washed and the signal is detected by measuring fluorescence at each spot. In the case of biotin labeled samples, the array is stained post hybridization with fluorescently labeled streptavidin. Laser induced fluorescence is typically measured with a scanning confocal microscope. The intensity of the signals on each spot is taken as a measure of the expression level of the corresponding gene. Transcription factor binding analysis. Microarrays have also been used in combination with chromatin immunoprecipitation to determine the binding sites of transcription. In this, transcription factors TFs are cross-linked to DNA with formaldehyde and the DNA is fragmented. The TFs of interest are affinity purified using either an antibody to the TF or by tagging the TF with peptide that is amenable to affinity chromatography. Example, a flag, His, MIC or HA tag. After purification, the DNA is released from the TF, amplified, labeled and hybridized to the array. This technique is commonly referred to as chip for chromatin immunoprecipitation on a chip or microarray. The TFs often bind quite a distance away from the genes that they regulate. The design of the array and the size distribution of the fragment length are interrelated. In bacteria or yeast, the intergenic regions are fairly small and the small arrays used for gene expression work can be applied to chip. For mammalian genomes, the intergenic regions are large and the TF often bind many kilobase pairs away from the gene of interest. Hence, for mammalian genomes, oligoarrays with oligos spaced evenly across the entire genome are typically used for chip experiments. Genotyping. Microarrays have been widely used as single nucleotide polymorphism genotyping platforms. Several alternative approaches have been used to detect these SNPs, but the most commonly used are allele discrimination by hybridization shown in figure 4a as used by Affymetrix, allele specific extension and ligation to a barcode oligo which is hybridized to a universal array, the Illumina Golden Gate assay as shown in figure 4b, or approaches in which the arrayed DNA is extended across the SNP in a single nucleotide extension reaction, arrayed primer extension assay as shown in figure 4c, or the Infinium assay of Illumina as shown in 4d. Allelic discrimination by hybridization suffers background due to non-specific hybridization in complex genomes. In order to reduce this background, Affymetrix developed a PCR based approach to reduce the genomic complexity. SNPs are selected for their assay to be between restriction sites that are less than 1 kb apart. Genomic DNA is fragmented with a restriction enzyme, end repaired, and adapters of PCR are ligated to the fragments. PCR is performed under conditions that selectively amplify products of less than 1 kb in size. This method reduces genome complexity by approximately 50-fold and results in a corresponding increase in signal to the noise on array. Both the Affymetrix and the Illumina methods of SNP genotyping have been highly successful and are highly used. Today, SNP arrays capable of detecting more than 1 m different human SNPs are available from both vendors.